So today, what we're going to do is start talking. All right, guys, listen. So we're going to start by talking about some general characteristics of cells and just an overview. Then what we're going to start to do is talk about the parts that are actually within cells, called cell organelles. And once we're done with cell organelles, we'll talk a little bit about some processes, some things that happen inside of cells that are important to know about. So first of all, you guys have an overview. We talked about it in our living things section. But basically cells are what all life is made out of. They're the basic unit of life. And they're found in all living things. The definition of a living thing is something that's made of cells. So it wasn't until 16 and 1700s that scientists really started to understand cells. Because what was necessary first before scientists could really start thinking about cells? Richard? Yeah, lenses, microscopes. Because cells are generally so small, they require a microscope to see any detail. And so it wasn't until the technology of lenses and microscopes was really refined that scientists were able to start seeing cells. So the first person that's credited with identifying cells was Robert Hooke. Robert Hooke was using a microscope to look at a variety of things. One of the things he looked at was cork. Okay? Cork. Well, this is a cork board. I have some cork somewhere. I'll look for it. But cork is, where does it come from? Like a natural, like a cork in a wine bottle? It comes from, a plant. It comes from a tree. It comes from the bark of a tree. And cork is not a living cell. What Hooke saw was not alive, but it was the remains of some plant cells. Okay? He saw that it was made of all these little sort of boxes, and so he called them cells because they looked like small rooms, like a cell, like a jail cell, and so he called them cells. It was another famous scientist that saw the first living cells. Anton van Leeuwenhoek is a famous scientist. He saw living cells. He looked at a variety of things. And in 1675, he saw in pond water these tiny little creatures that you couldn't see with your naked eye, but you could see with the microscope. And so he saw that there are actual living creatures there that were microscopic. They were a single cell, and they're things like amoeba or paramecium. He saw living cells in water, in blood, in sperm, and he noticed that these were all made of cells. And Leeuwenhoek was kind of interesting. He was very secretive. His first job was as a draper, person that made like curtain drape things. And one thing he did is he was looking at the material and the threads to make this drapery material, and that's how he got interested in microscopes. And he actually made the most powerful lenses at the time, but he was very secretive. Other scientists would have loved to make them microscopes or see how he did it. But he was so secretive, he wouldn't tell anybody. When he showed people his microscopes, he would save the best ones and not show anybody, and just show them his inferior ones. And they didn't look at all like a microscope that you might imagine. They didn't look like our microscopes or even these older microscopes. This is what one of Leeuwenhoek's microscopes looked like, this sort of weird piece of metal. And what it is, this little area right here has a tiny glass lens in it. And you would hold this thing up to your eye and look through that hole, through this tiny lens, and the sample you're looking at, you put on the top of this little needle sort of thing. And then you could use those knobs to adjust the focus or raise or lower the specimen. But that's the sort of lens he was using. It was just a single lens, so not a two-lens microscope like we would have. But he made these lenses so good that they could magnify up to 300 times with just a tiny little lens. And so his microscopes were really good. This is a little, I don't know if we should watch this.
Let's watch it. Let's watch it.
Yeah, we should.
We should watch
So this is, we'll see. I guess.
It's crazy that it's like
everywhere.
technology's advanced so far, but back in the one of the very first microscopes could
Yeah, just as good as the microscopes we're using.
Even
Yeah.
back, like the ones we use only work a measly 40. Great job, guys. I'm looking at you, right?
I love watching.
Everything that you can actually see with your eye is just the smallest sliver of life on this earth. Most of life is invisible. We still have this idea that we're the most central feature of Earth, and it's the humans that are the bystanders. The microbes are doing the work.
Wat doe je als je dingen ziet wat niemand ooit heeft gezien? What do you do when you see things that no one has ever seen before? L-A-Y, lay, u, then, they pronounce it with a V, Hooke, Leeuwenhoek.
Anthony van Leeuwenhoek. He was a haberdasher in the city of Delft in the Netherlands.
And why his curiosity found an outlet in microscopes that is just lost to history, we really don't know.
The quality of his microscope was superb. He made some 500 of these small instruments, and only a few of them he showed to visitors. He never told anyone how he made his lenses.
Robert Hooke in England, he wrote this wonderful book, Micrographia.
First observations of the small world with lenses.
One of the first things that Leeuwenhoek did was look at things that Hooke had looked at. There was a stinger of a bee, leg, I believe, of a louse, singular of lice. But he saw some things that Hooke didn't see because his lenses were better. It was summertime, it was August. The days are so long that you get a lot of algae growth on water. He called it green clouds. Curious again, he had what he calls a glass vessel, a jar probably, and he filled it with the water. The next day he put it under his lens, and what he saw was green streaks. Among this was all these little animals. And these things were a whole lot smaller, like 1,000 times smaller than anything he had ever seen before. And I think the line is, I confess I could not but wonder at it.
Leeuwenhoek called them in Dutch dierkunst. A dierkunst, that's a diminutive of the word deer,
Deer, D-I-E-R.
and which is the Dutch word for animal.
What Leeuwenhoek called was little animals.
This was all so new. The word microorganism did not exist at the time. The word bacteria is from the 19th century.
And that strikes me as Adam in the Garden of Eden, who in Genesis named all the animals. It was just a brand new world. And he was the first person in it.
He wrote a letter to the Royal Society, one of the first organizations to practice experimental science.
And they're going, oh my heavens, what is this?
At first they didn't believe it.
Finally, the other members of the Royal Society were also able to see it. And the rest is history.
And so he discovered many things.
Sperm, red blood cells, protozoa and bacteria.
which nobody had ever seen before.
He is the first person to see everything he looked at for 50 years.
Van Leeuwenhoek wanted to see these things. Well, he saw them. But now we get most of life is microbial. If you look at the tree of life, only this tiny little part is every single thing you've seen. Every higher organism is covered, inside and out with bacteria. And humans would not be alive if these little 24/7 partners weren't giving us all of these genes and proteins that our own genomes don't encode. And they have all kinds of fabulous behaviors. Vibrio harvii is a marine bacterium that looks like a sausage, and it's very fast. Vibrio means vibrate. And what is amazing is that if one watches them go from a single cell to a number of cells, all of the bacteria in unison start glowing in the dark. By studying this bioluminescent organism, we discovered that bacteria can communicate using a molecular language. We used to think that social behaviors were the purview of higher organisms. What we now understand is that bacteria were probably the first organisms on this earth to ever communicate with one another. We're always looking at an unknown world. We're driven by our ignorance, and we're driven by the idea that the world must be more complex than what we understand right now. And that's enough inspiration to do an experiment.
All right. So just a little bit about Leeuwenhoek and about, like they said, the majority of life is things that we can't see. All right. Colin?
If it says in, during this video talking about how he was the one to really make good microscopes and stuff like that, did they have just worse versions? Cause
Yeah, they had other microscopes.
1665, the Robert
Yes.
Hooke guy looked in the first one. Well, saw the cells under the first one, but then it said 1675. It said
Yeah, the quality of the lenses were far inferior to what Leeuwenhoek was able to do. All right, so after scientists started to discover cells, and there's a whole host of scientists that added to our knowledge about cells, what we have is something called the cell theory. Cell theory is sort of a set of statements that we know governs living things, and it's related to cells. So first off, cell theory says that all life, anything that is alive, is made of cells. One or more. Cells are the smallest form of life. Like we said, an amoeba is a single cell. A bacteria is a living thing, a living organism. It's all the characteristics of a living thing, but it's only a single cell. Tiny, tiny little cell. And the last part has to do with where cells come from. You can probably guess the answer to that. Where do they come from?
just other cells.
Other cells. Cells come from other cells. And so within cells, there are smaller parts. So what do we call inside of our bodies the parts that have certain functions, certain jobs to do?
Organs.
Organs. In a cell, there are the same things, small parts within cells that have a certain job to do, but we don't call them organs, we call them organelles, small organs, basically. And we're going to be talking about these organelles over the next couple classes. And what we're going to do is sort of talk about what they look like, what their function is within the organism, to help us understand how cells actually work. Colin?
What is the bare minimum magnification needed to see the cell?
It depends because cell size varies. There are some cells that are pretty large, like a human egg cell. one of the largest cells in the body that you can see without a microscope. But that's rare. Most are, you need some level of magnification just to see that it's there, and to see any detail, you need greater magnifications.
Are our lenses right now better than the longer?
Yeah, we have better lenses than that.
Longer.
They actually figured out, they do know how he made them. He would melt glass into a liquid, drop tiny little droplets of the glass into water, where they immediately cool and form a tiny little spherical lens that was very clear. And most other lenses at the time were made by grinding glass, like using grinding rough materials to get the glass into a certain shape. And so this is a completely different means of creating a lens. No, in that case it did not.
I mean, I bet you probably had some that weren't like perfect.
Yeah,
I bet you
yeah.
there's a lot of trial and error.
Yes.
Just because like with anything.
But nobody was even using that method to try to make one. All right, so organelles, parts of cells with a certain job to do. So in our notes, we're going to talk about kind of like a generic cell. And actually, cells within our body are all different from each other. A nerve cell is very different from a skin cell, which is very different from a blood cell. But we're sort of talking about these sort of just general characteristics of cells. Most cells in our body don't look like what we're about to talk about. We're also going to split these cells up into two broad categories, animal cells and plant cells. Now again, that's kind of artificial. There are other types of cells. And the cells within plants and animals look different from each other, but this will just help give us sort of a guide to talk about the cells. So, can you guess, these are two examples of cells. Can anyone guess which is the plant cell and give me a reason? Walker?
The green one, the
Green.
Okay.
That's big facts.
The green one, because it's green. What does green have to do with anything? Plants are green?
Lots of them are green.
This plant's not green.
I said
Lots.
lots.
Lots. Other ones are.
Lots.
So why are many plants green?
Because the cells in them, obviously.
And they have a scientific answer? Ashley?
With the chlorophyll.
Yeah, because they have chloroplasts, which we'll talk about.
Because the mitochondria is the powerhouse of the cell.
All right.
And the nucleus is the brain.
So plant cells are generally, sometimes they're green, depends what part of the plant they come from. A cell that comes from the roots of a plant is generally not green. Okay, a cell from the interior of a tree trunk is not green, but sometimes they are green. Their shape is different. Okay, that's another important factor. They're generally sort of rectangular or angular. They have also this hard outer shell called the cell wall. It's only found in, well, it's found in also fungi. It's also found in bacteria. So this is not really the best way to phrase this, but in terms of plant versus animal, only the plant cell has a cell wall. And the cell wall of a plant is made out of this rough material called cellulose. It's a large molecule, and it's a sort of tough outer layer. And it helps to protect the plant cell, helps to give it its shape. One interesting thing about the cell wall is that we humans cannot really digest cellulose very well. Okay? It's a very hard material for many organisms to digest. So when we talk about getting your fiber, okay, that's cellulose. Because we can't digest it. Therefore, what happens to it in our digestive
It
system?
pushes it.
It just goes right through. Yeah, eventually out the other side. There are some organisms which can digest cellulose. Okay? For example, in termites, they have a bacteria that lives in their digestive tract that can digest it. So there are organisms that can digest that cellulose. But this outer layer, this cell wall, that's found in plant cells, not animal cells, and it's actually not a living part of the cell. It forms and then it's just a shell. Okay, it's not flexible, doesn't move very much once it's formed, and it sticks around for a while after the plant cell dies. So therefore, when Robert Hooke was looking at cork, he was looking at the cell wall that was still there from once the plant was alive. So the next layer, just inside of the cell wall in a plant cell, or the outermost layer of the animal cell, is a living part, and that's called the cell membrane. Okay, so the cell membrane is a thin layer that's around the outside, and it's really important because it does several things. It determines what material can enter the cell and what cannot. It's the boundary between the living cell and the outer portions that are not part of the cell. It helps cells communicate with each other. It does a lot of different things. And I will tell you right now, just like I have in a past quiz, I'll ask you a question on an upcoming quiz. It's going to say, what is the outermost living layer of a plant cell? And many of you may get this question wrong because you will say cell wall. But it's not living. The cell membrane is the outermost living layer of a plant cell. The space inside of the cell, okay, is called the cytoplasm. Cyto means cell. Plasm kind of means like shell. And so the cytoplasm of the cell holds the other organelles. Things move through the cytoplasm. Like a, I don't know, did you ever have like maybe someone in your family nowadays makes a jello mold that's like jello, those like chunks of fruit kind of stuck in it?
I've seen it sometimes.
It's kind of like, oh, it's not really that much like the cytoplasm. The cytoplasm is more liquid, but the organelles would be like little chunks of fruit that, you know, your aunt put in the jello.
So you know like coffee? Alright, let's
Alright.
talk about skeletons.
So, if we look at this diagram, this shows two animal cells, and we can see a few different organelles in them. We can see the cell membrane around the outside. We can see this dark spot in them is the nucleus, and then the white area represents the cytoplasm of these cells. The nucleus. Nucleus is often the largest organelle that you can see inside of the cell. And it's generally roundish in shape, and it's made of several parts, actually. And there's one important thing, well, there's several important things, but the most important thing that's found inside the nucleus is the DNA, the chromosomes. The nucleus controls the activity of the cell because it contains the DNA. The DNA is what makes the cell. It's the DNA that codes all the information. The DNA in your cells makes you who you are. And so that DNA spells out how to build the cell, how to build the organism, how it functions. So sometimes people say it's kind of like the brain of the cell. Even though really the cell doesn't think. Okay? The nucleus itself, it holds the DNA, which is an important part of the cell.
Like on the page before that, with the two animal cells,
Yep.
what is like in the area where there's just nothing? Like what is just the blank space there? What is that,
That?
made of?
This?
No, like,
Here?
yeah, what is
Probably
that?
just water. Or it could be, yeah, it's just water probably. All right, so this is the nucleus as a whole. And in an electron microscope, it just sort of looks like a circular organelle. The outer layer is called the nuclear membrane. Okay, it surrounds the nucleus, has these holes in it, which allow some things to move through and other things cannot. The DNA is actually stuck inside of the nucleus. It's too large to fit out through these openings. Check?
So the nucleus is the brain of the cell?
No, it is not the brain. The cell doesn't have a brain. It's kind of like the brain of the cell.
So
Notice the quotes, Jack. Do you notice the quotes? So it is not the brain. The cell doesn't have a brain. The cell doesn't think. So the nuclear membrane surrounds it. Inside are these chromosomes. Chromosomes are the genetic information of a cell. In your cells, in typical body cells, humans have 46 chromosomes. 23 of them you got from each parent. And they contain your genetic information. Your chromosomes contain your DNA, which has your genes, which makes you who you are. And then inside of the nucleus, there's also this area that's a darker color often. It's called the nucleolus. That means little nucleus. And the nucleolus makes another organelle called the ribosome, which we'll talk about. The chromosome in certain times looks like an X. Other times it unwinds into sort of a loose ball of string. But that's a chromosome. Holly?
Okay, coming back to what Jack said about how the nucleus of the brain is up.
He didn't, yeah, and I corrected him and told him that
But
it's
the
not
cells
really brain.
think like all living things. They also need brains. So wouldn't something need to like have a brain of its own to control the brain?
Yep.
No, not really.
What?
So, the ER, the endoplasmic reticulum,
Our brain named our
is abbreviated oftentimes ER. And the endoplasmic reticulum is this series of tubes and membranes that winds from the nucleus to other parts of the cell. And it's important for transporting material within the cell. One way to help people remember is if you got hurt, you would have to be transported to the ER, emergency room. The ER's job is to transport things.
Endoplasmic reticulum. How do you say the last term?
Endoplasmic reticulum. And there's two versions of this in the cell. Rough ER looks like this. Looks like chocolate chips on it. And then the smooth ER doesn't have those chocolate chips. So these chocolate chips on the rough ER are actually another cell structure called the ribosome. Ribosome is a tiny, tiny little part of the cell that's job is to make proteins. Almost all of living things are made of protein. And it's these ribosomes which build these proteins through the cell. I tell people to remember, like, ribs, meat, you know, the thing you would grill, ribs, made of meat, which has a lot of what nutrient? Protein. So ribosomes make protein.
He said.
Grill or whatever. You tear apart the ribs of an animal and gnaw the flesh in
Oh, hold up,
between.
are you a vegetarian?
Yeah.
Hold up, you can't say that.
I can say it, I just wouldn't eat it.
That's
Um,
Why do you talk about
That's
Ribosomes.
like me saying, I'm
Some are, all right, don't worry about it. Some ribosomes are found stuck to the ER on the rough ER. Other ribosomes are just floating around in the cytoplasm on their own. The Golgi bodies. You have questions? Ashley?
How come some of the ribosomes have how come some of the ERs
Oh.
have them, some of them don't?
Some of the ER make certain proteins, and the other parts of the ER are responsible for making other material. That they don't make proteins, they make fats and lipids and other things, so they don't have those ribosomes. Good question. Picture? Raphia, oh, the little cartoon?
Yeah.
Hey ladies, how about a date?
Hey ladies, how about a date? Oh man, oh man, he gets all the girls.
Like, why?
Smooth, because he's smooth.
Smooth?
He's so smooth, he gets all the girls.
Is it like
He's a smooth ER.
God, don't you
If it takes this much
No wonder why.
explanation, it's a bad joke. All right, Golgi bodies.
There's 10
The Golgi bodies are like the post office of the cell.
No.
What they do is they take these proteins, package them up if they're going to be leaving the cell. And it sends them on their way to exit the cell. Sometimes I say they look like a stack of delicious pancakes with delicious drops of grade A amber 100% pure New York State maple syrup.
Okay, that's better.
So you can see these little stack of pancakes and those drops of syrup. Those are the Golgi bodies.
I would love that. It's like some
And so the Golgi bodies package up these proteins, put them into these little bubbles called vacuoles, and send them to leave the cell. Anybody familiar with the band Fish?
Yeah.
They have a song that mentions Golgi bread.
The band?
Oh, I wish.
How many
You're a
people are
vegetarian.
familiar with the band Fish?
We'll just two.
One, all
Oh yeah,
right.
the names are
Blind
blind fish.
Yeah.
Just two.
Just one person? All right,
Wait.
I'll play a song for you before class.
No, obviously.
Two of us.
Math.
All right, let's finish up this last slide, then we can
Waste your
finish
time.
up.
All they do is forget about.
All right.
Vacuole.
Vacuoles. Vacuoles' job is to store things. They're these little bubbles within the cell that basically can store water or other materials. Plants often have a very large vacuole in the middle of the cell. Animals usually have smaller vacuoles spread throughout the cell. So this thing looks like an ice cube. That's a large vacuole that's inside of the plant cell, whereas the animal cell, just little circles are the vacuoles. And they store things. And then finally, the last organelle for today are the lysosomes. Lysosomes, this is also a vacuole in the plant cell. Lysosomes have enzymes in them for digesting material. So anything that needs to be broken down within the cell, the lysosomes can digest those parts, sort of recycle them. If the lysosome in the cell just breaks open and spills the enzymes into the cell just digests from the inside out and is destroyed. White blood cells, which find and engulf bacteria and stuff, they have many lysosomes in them because that's how they destroy the pathogenic cells. All right. So we'll pause there. Till tomorrow.